Welcome to problem set 22. Our problems today will be from the topic, the lipid bilayer. Before we get started, just a quick reminder that the worksheet to accompany this problem set video is available online through my website. And with that said, let's get started with number 1. Number 1. What portion of a phospholipid is hydrophilic and what portion is hydrophobic? So remember that the charged phosphate unit will be hydrophilic, that is water-loving. It will interact with the partial positive and partial negative charges on the water molecules. So the charged phosphate unit is the hydrophilic or water-loving portion of the phospholipid. The 2 hydrocarbon tails of the fatty acid will be the hydrophobic portion. The 2 hydrocarbon tails are hydrophobic. So if we use our cartoon diagram, which is the head, the phosphate head with the 2 hydrocarbon tails, then we would say that the phosphate head is hydrophilic, water-loving. It will interact with water molecules. And the 2 nonpolar hydrocarbon tails will be hydrophobic. That is, they will not be attracted to the partial positive and partial negative charges on the water molecules because they themselves do not have any partial positive, partial negative charge. They are nonpolar. So hydrophobic tails. I should write head here. The phosphate and the hydrocarbon tails. Okay. Number 2. Using the generic head and tail diagram for phospholipids, like what we did here, show how 2 layers will coagulate together to form the lipid bilayer, labeled the hydrophobic and hydrophilic regions. Okay, so for number 2, we're going to draw a portion of a cell membrane. We'll keep it kind of small, otherwise this problem will take too long. Remember that we have the extracellular fluid, that is the fluid outside of the cell, and that is aqueous. And we have the intracellular fluid, that is the fluid inside the cell, and that is also aqueous. And I'm only drawing a portion of the cell membrane. I'm not going all the way around just because I don't feel like drawing that many phospholipids, even if I am using the cartoon diagram. Okay, so remember that the phospholipids will orient themselves such that the hydrophilic, water-loving head, the phosphate unit, is oriented toward the aqueous environments. So that is toward the extracellular fluid. And toward the intracellular fluid. I'm going to put another layer here of those phosphate heads. Okay, and the hydrophobic, water-fearing hydrocarbon tails will coagulate together in the middle of the bilayer away from these aqueous environments. So I'll put 2 tails on each head. Kind of tedious. But you can imagine it's a lot faster than actually drawing all the carbons and everything out and all the phosphates. So a little cartoon depiction helps a lot. And then 2 tails on all of the ones aiming towards the intracellular fluid as well. Those tails are all nonpolar, so they coagulate together in the center there. Now remember that those tails can be saturated or unsaturated chains. They can be from saturated or unsaturated fatty acids. All right, so then the last thing we have to do, and let's do it in the blue pen, is label the hydrophobic and hydrophilic regions. So we know that these phosphate heads are hydrophilic. Phosphate heads are hydrophilic. Water-loving. That's why they orient toward the aqueous environments. We know that all of the hydrocarbon tails are nonpolar. They're hydrophobic. That's why they orient away from the aqueous environments and coagulate together in the middle of the bilayer. So the hydrocarbon tails are the hydrophobic regions of the bilayer. All right, so 2 sections labeled as hydrophilic, that is one facing the extracellular fluid, which is the phosphate heads, one facing the intracellular fluid, which is the phosphate heads as well, and one region is labeled as hydrophobic, that is the middle region, which has all of your hydrocarbon tails. All right, let's go ahead and go on to number 3. What molecule adds more rigidity to the otherwise fluid lipid bilayer? Draw its general structure. Okay, so number 3, that molecule is cholesterol. And remember, it has a nice rigid structure because it's composed of those 4 fused rings of alsterols. And to draw that general 4 fused ring structure, we do a 6-membered ring attached to another 6-membered ring, attached to a third 6-membered ring, and finally a 5-membered ring. Okay, so 6, 6, 6, and 5, and that structure is kind of plate-like and rigid. All right, let's erase here before we go on to number 4 and 5. Number 4, which of the following uses energy to maintain different concentrations across the cell membrane? A, simple diffusion, B, facilitated diffusion, C, passive transport, and D, active transport. The answer is D. Only active transport uses energy to maintain different concentrations across the cell membrane. So let's write that up here. D, the answer is D, active transport. Notice that A, simple diffusion, and B, facilitated diffusion, those are just 2 types of passive transport. And passive transport does not use energy because passive transport relies on diffusion. That is, the natural tendencies of particles to spread out and flow from a region of higher concentration naturally to a region of lower concentration until both sides have reached an equilibrium. Okay, so that does not take energy. That relies on particles' natural tendency to spread out. Active transport takes energy because active transport is maintaining different concentrations across the cell membrane, and that takes energy. All right, number 5, last but not least, explain how facilitated diffusion and active transport differ. Okay, so remember that facilitated diffusion occurs when a molecule is too large to undergo just simple diffusion through a protein channel. And so in facilitated diffusion, that larger molecule binds to an integral protein, causing that protein to change its shape and carry the molecule over to the other side. Now, this does not take energy because it still relies on the natural tendency of molecules to move from a region of higher concentration to a region of lower concentration until they equal out. And that's a natural tendency. It doesn't require energy to do that. Okay, so let's go ahead and write a little summary of facilitated diffusion, and then we'll talk about active transport, and then we'll get to the final answer of how they are different. So facilitated diffusion. In facilitated diffusion, a molecule that is too large to diffuse through a protein channel binds to a protein, inducing a change in that protein's shape. This change in shape carries the molecule to the other side of the bilayer. Carries the molecule to the other side of the bilayer. Remember, this does not require energy because we're still relying on the natural tendency of molecules to progress from higher concentration, to move, I should say, from a higher concentration into a lower concentration. So, still move from higher concentration to a lower concentration. So this is just a type of passive transport. Okay. So active transport we'll talk about next. And let me underline those words. Okay. In active transport, energy from the hydrolysis of ATP is required to continuously pump particles and maintain different concentrations. So rather than naturally allowing particles to just flow from higher concentration to lower concentration until they equalize, in the case of active transport, we're continuously pumping particles to maintain the different concentrations on each side of the lipid bilayer. Okay, so it's right here. In active transport, energy is required to continuously pump particles across the bilayer in order to maintain different concentrations or a concentration gradient. Okay, so the key difference here is that in facilitated diffusion, we're talking about the natural tendency of molecules to flow from higher to a lower concentration until they even out on both sides. This does not take energy. This happens naturally. In active transport, we're talking about the use of energy to continuously pump particles across the bilayer and maintain different concentrations. So the energy here is key. Make sure you understand why active transport requires energy. It's maintaining a concentration gradient. And why facilitated diffusion does not. It's using the natural tendency of molecules to spread out from a region of higher to lower concentration until things become equal. Okay, so this will conclude our very short problem set 22. I hope that you will consider tuning into our tutorial number 23. Thank you very much.